Hello everyone, a very good afternoon to all of you. I hope you all are good. So we are done with the unit 2.1 and 2.2 from the Socio-Cultural Anthropology, and today we are basically giving you a brief introduction about the physical anthropology, because it is also one of the important aspect whenever you are trying to cover your syllabus for anthropology paper 1. You know already that in terms of a basic introduction I had provided you in terms of orientation, where I had told you that physical anthropology, it is about 24% of the total syllabus for anthropology, and it is the part of the paper 1. Many of the students, whenever they are thinking about taking the anthropology as their optional, they used to feel the concern like anthropology is all about science. So today I just want to burst that myth or that concern, because physical anthropology or this biology part is completely same or similar with your general science in the prelims as well as your science and tech for the GS paper 3. In today's lecture, we would be taking up the unit 9.4, but before that, I will be giving you a brief overview about the physical anthropology and that part. Before that, Hanji, good afternoon Navya, good afternoon Hari. I hope you all are good. Okay, so before that, just tell me if you have any doubt, any query related to the previous lectures. If you have, you can just share your queries in the comment box. And now let me give you the overview about the physical anthropology. Personally, if you would ask me, I would say among the all parts of the anthropology, as you know, socio-cultural anthropology, physical anthropology, then Indian archaeology, Indian anthropology, and tribal India. Among all these five, the most interesting one and something that would excite you and will help you to know about the human beings as well as the live anatomy of the human being, it is none other than the physical anthropology. Another thing in terms of physical anthropology, like in the previous PYQ discussion, I had told you the way how you can approach the answer related to the socio-cultural anthropology, but the same way is not going to be there for physical anthropology. I will let you know the answer writing art for writing the answer for this physical anthropology part as well. We are launching our course in the offline as well as in the online live mode this time, and our batch is going to start from 10th of June. If you want to inquire for the batch, or if you want to know about the basic details, you can get in touch with us. There is a Google form available in the description box. You can just fill that form and our team is going to connect with you. Other than this, there is number available in the description box. You can directly connect with that number as well. Okay. Do you have any doubt? This is my first live lecture to watch. Okay, Navya, that's great. So today only we are taking up the physical anthropology part. So do not hesitate. Even if you are not able to understand anything, do not hesitate at all. Ask me. So I am going to repeat it that. Very good. That's great, Navya. Okay, so whenever we have to introduce the physical anthropology, this is the first picture. And the first picture, it is showing that we as a human being, we have introduced some sort of changes, and these changes, they are some transitional changes, and we are changing our shape from this to this, and this is something based upon over a period of time. Fine. So that's why you would feel, oh, it's so much difficult, like we have to read so many theories how the humans, they like transitioned from the apes or the monkeys, how this change is going to proceed. Fine. So this is not the case. There is only single one unit that is going to introduce about it, but other than this, we are going to know about the anatomy of the human being, of the man. Fine. In terms of the physical anthropology, first of all, the only thing I used to introduce about the syllabus, and the only thing that I used to tell you about the syllabus is this, that you need to first introduce yourself with each and every part of syllabus. today we are starting with the physical anthropology. First, I want to tell you the part of the syllabus. And whenever you are starting preparation for any subject, be it your GS subject, be it any part of the anthropology or any another stream. So the first and the foremost thing you need to know is about the syllabus, and that should be in the raw format. Raw means as being specified by the UPSC. So in terms of the physical anthropology, there are some specific units. As you know, it is nothing but the part of paper 1. And whenever you are starting preparing your notes, this simplified thing should be sorted in your mind. Okay? This simplified thing should be sorted in your mind. So, physical anthropology is constituting the paper 1, and out of this total syllabus, it is around 24%. In terms of weightage. Now, unit 1.4 to 1.7. Then, unit 9 to unit 12. These are the units that's being part of the physical anthropology. So, you will see first the syllabus for the paper 1, and in the paper 1 syllabus, there is these units, unit 1.4 to 1.7, then unit 9 to 12. Unit 9 is having 8 subpart, then unit 10, then unit 11 is having 3 subpart, and then unit 12. So that's how the total composition for physical anthropology is being specified. Okay. Now there is unit 9.1, 9.2, 9.3 and 9.4. In today's class, I am starting with the unit 9.4. Reason? Because I just want to introduce some aspect of the physical anthropology, and in terms of introduction, this is the unit that we should take up for the very first time. Once we are done with this unit, then we will be coming towards 9.1, 9.2 and 9.3. Reason? Because the syllabus as given by the UPSC for the unit 9.4 is going to be a base for the further units. Okay. So therefore, in today's class, we are taking up the unit 9.4. Another thing, as from the lecture, you know very well that whenever we are starting with the unit, any unit, first and foremost thing, we need to write up the syllabus in the same words as being specified by the UPSC. Then to divide that syllabus into various parts, then take up one part and do that part comprehensively. Fine. So in the same way, we are taking up the unit 9.4 and the syllabus of the unit 9.4. So this unit 9.4, it is provided as the chromosomes and the chromosomal aberration in man methodology. This is something that is being specified by the UPSC. Now, this is being divided by me into 4 parts. The first part, they are asking about numerical and structural aberration. Then they are asking about the sex chromosomal aberration. Then about the autosomal aberration. Then genetic imprint in the human disease, genetic screening, genetic counseling, human DNA profiling, gene mapping and genome studies. In today's class, I would be taking up this part. So first we will try to know what do we mean by the word aberration. But before knowing about the word aberration, we would be explaining about the word chromosome. Once we are done with the basic explanation of the word chromosome, then we would be explaining the numerical and structural aberration first, and then sex chromosomal aberration, Klinefelter, autosomal aberration. So we will try to know what do we mean by these words, autosome, what do we mean by the word sex chromosome, and what are their applications. So first, I would like to tell you the basics of the chromosome. Quite possible there are so many students who are having the biology background, so they know already basics about the chromosome, but still, because I am considering that you are starting from the scratch and you are the beginner, therefore I will let you know the basic details of the chromosomes. Okay, fine. Do you have any queries? No? That's great. Okay. Now, as starting with the numerical and the structural elaboration, now let's start. So, the first and the foremost thing that I am going to introduce you here is the cell. Cell means what? Cell, it is the smallest functional unit. So, it is the most smallest functional unit and it is the functional one. I mean, it is the smallest unit as well as it is the functional one. Functional means every single cell is independent. They are having so many components and these components, they are working for that. I mean, this is something which is you have read in your NCRTs, that is there is mitochondria. Okay, you are going to tell me what do we mean by the mitochondria? What are the kind of function this mitochondria is doing? So what do we mean by the word mitochondria? Ali, quickly. Okay, so basically the mitochondria is considered as the powerhouse. Why? Because mitochondria is taking up the oxygen and from the oxygen they are converting this oxygen into the ATPs and 38 ATPs they are taking up the form of energy and this energy is nothing but that's why it is being considered as the powerhouse. So power is coming from the energy and energy is being produced by whom? By the mitochondria. So this is the basic composition of the cell. Every single part of this cell, they are working. And these parts, they are working for what? They are working for the nucleus. Now, I would be explaining the cell and all the component of the cell in a very comprehensive manner. But I will be explaining the cell in the lecture unit 1.7. Okay, so I would be explaining each and every component of the cell, the DNA replication, then DNA duplication, some DNA based error in the lecture unit 1.7. Fine. In today's class, I would be considering about what? The nucleus. Now you know very well that human body is nothing but it is a combination. Whenever we are clubbing together the cell, then a human body is formed. So therefore, they are clubbing together and because of their clubbing, tissues are formed. When tissues are clubbed together, then organs are formed. And when we are clubbing the organs, then human body is formed. This is something like a very 8th standard phenomena, but that's how our body is being formed. Because we want to understand about the form of the body, therefore we are taking up the cell into the consideration. So the detailed explanation would be there in the unit 1.7. Now for today's lecture, what I am going to take is about this part. So all other component of the cell, they are working and they are working to provide each and every, like just to fulfill each and every requirement of this, the king of the cell, the nucleus. So this nucleus is being considered as the king of this cell, and if the nucleus would be living, the cell would be living, and if it is dead, it means completely cell is out of life. Fine, it means this nucleus is something which is important. Now, what I would be doing is, I will have a detailed microscopic view of this nucleus, and from this nucleus, I will get something which I required for today's lecture, chromosome. Now, let's see. So, this is basically the cell. The cell is having what? The cell is having the nucleus. So nucleus, when we are going to dive into the nucleus and we'll have the microscopic view, I will find out this type of structure. Okay, so just wait for a while. Let me give you the overview. So let us suppose this is my cell. In this cell, I am having I am having this as a nucleus. Okay. Then I will be dive into this nucleus and will have a very microscopic view. I will get what? I will get this type of structure. Okay, one more thing. Actually, I will get this type of structure in terms of a chromosome. But for studies perspective, we used to have this type of structure. Okay, so for the classes or for the study perspective, we will have this type of structure. So these are what? This structure is nothing but it is the chromosome. Now, because these chromosomes, they are arranged in such a way, like just try to understand. These are the chromosome and they are like arranged in this way, therefore like they are arranged in this way, therefore I can see from far away this condensed structure, and when I will be zooming it out, I will get this type of structure in terms of a chromosome. Okay, so I hope you can understand the basic part of the chromosome. Now I have to study from this chromosome the basic genetic material. Genetic material means something which is considered as the identity of a human being. Now you would say, ma'am, our Aadhaar card is our identity. Although Aadhaar card can be forced, but still we used to say like this is my identity, my Aadhaar card. But here try to understand. Aadhaar card is in terms of this identity. When I have to see the basic anatomy, the internal structure of a human being, so I can understand the internal structure of a human being from the DNA. DNA is the genetic material. Now, when I would be taking up this chromosome, so let's see, I am taking this chromosome, and chromosome, they are having these types of arms. So this one and this one. Because I want to understand more, therefore I am going to zoom more. And after the zooming, I will get this structure. I mean, I will try to stretch this arm, and because I'm trying to stretch this arm, I can get this type of structure. This loosely type of structure. Now this structure would help me to understand about the chromosome. Fine. So from here, I can understand that the cell is having the nucleus. Nucleus is having the chromosome. Now the chromosome is here in terms of joining together the DNA. And the DNA is having my gene. Fine. So now what we are going to do is we are going to understand about the chromosome and the structure of the chromosome. So now let's try to understand. I hope you have understood so far. Okay, now okay, so try to understand, this is what? This is the chromosome. Now, one cell is having 23 pair of chromosome. Just try to understand this thing. Here, I am taking up one chromosome and will try to make you understand the basic structure of this chromosome. Then we would be adding up all the chromosome, and then we would be adding up all 23 pair of chromosome. Fine. So I will go step by step. You just try to understand each and every part. Another thing, in terms of the physical anthropology, there is going to be a difference. In the socio-cultural anthropology, we were trying to add upon more and more name of the anthropologists. We were using more and more example. We were adding up so many case studies as well as current things. Let me assure you, current things, they are again going to be part of the unit 9.4. But here in the unit 9, in the physical anthropology, the name of the anthropologist, we are going, like rarely we are going to use any name. In terms of example, rarely we are going to use an example, but here we will use up the information, the facts and some factual arguments. Okay, so this is what we are going to have. Now, this part and this structure is talking about one chromosome. Now, I would be helping you just to name about the basic thing, and then like we'll take up to the advanced level. So, because it is the chromosome and it is something that is condensed one, and the same is going to be on this side. Okay, so this is something specified in terms of chromosome. Fine. Now, let's try to name it. Okay. So, this complete structure is called as what? This complete structure is called as the chromosome. This complete structure is specified as chromosome. Now, in this chromosome, this one part, this one side, consider it like a flower. So, this flower is attached like this. Okay, so kindly consider this one as one arm and this one as another arm. So, it is like this in this structure. It is in this way, it is like a chromosome. Here you can understand this is the chromosome. Now, this side, this V-like structure, this side consider as chromatid. This side again consider as chromatid. When I am attaching these two chromatid, I am having what? One chromosome. Okay. So now let me give you the another view. Let us suppose this is the one chromosome. Now this part, let us suppose it is A, it is B. This A part and this B part. A part and B part, they both would be specified as what? They are called as chromatid. Chromatid is considered as the condensed form of DNA. Therefore, from this perspective, you can understand that this part, it is specified as chromatid. It is specified as what? Chromatid. Now, chromatid is nothing but it is structure of DNA, and DNA is packed together and condensed in such format, condensed in this format. Condensed in this format, and this is specified as what? Chromatid. It means you are going to answer me. I'm having one chromosome. So one chromosome is having how many chromatid? Just answer me. One chromosome is having, I'm talking about one chromosome. So one chromosome is having how many chromatid? So you are going to answer me. Do not hesitate. Either it's going to be right or wrong. Just answer. One chromosome is having how many chromatid? So I'm waiting for your answer. Try to answer. Okay, so Swati is saying it's 2. Okay, Navya is again saying 2. That's great, very good. It means one chromosome is having 2 chromatid. Now, here we are having one pair of chromosome. What? We are having one pair of chromosome. So, answer me. One pair of chromosome is having how many chromatid? One pair of chromosome is having how many chromatid? So one pair of chromosome is having, I'm waiting for your answer. It's having 4. Very good. Now, try to understand. This chromatid is nothing but it is most condensed DNA. Okay, now this most condensed DNA is now being opened and it is being stretched. So when we are trying to open it and when we are stretching it out, then it is being formed in this format. And this, it is also condensed, but it is less condensed than chromatid. So it is being specified as what? Chromatin. So it is being specified as what? Chromatin. So chromatin is part of chromatid, yes or no? So just answer me. Chromatin is part of chromatid, yes or no? Yes or no? Yes, very good. Now there is no difference in the chromatid and chromatin, and unless then the structure. So chromatid, it is the most condensed, chromatin it is less condensed. So it is nothing but it is the less condensed DNA. Fine. Now then if I'm taking this part, this double helix part, so this double helix part, it is nothing but it is being specified as what? It is being specified as the DNA. So this double helix, double stranded structure which is being opened, this structure is called as what? DNA. And we are trying to study the chromosome because we want to study about the DNA. Now, complete DNA is not having information. DNA is somehow, like somewhere it's having some sort of information, somewhere there is some sort of junk material being attached together. So just tell me one thing. You must have seen some very Pinterest type of packaging. So the packaging which includes so much of the paper and paper cuttings and all, so that your package material, if that is fragile, can be protected, yes or no? If you have ordered any makeup or any fragile thing online, so you have received the package and that product is wrapped together with some loose parts of the paper cutting, yes or no? Just tell me. And the reason behind that is so that they can protect the product, they can deliver the product. In the same way, complete DNA is not having your genetic material. DNA is being packed together junk material as well, so that they can protect your gene. So therefore 99% of the DNA is junk DNA. If you have ordered some makeup or anything from the Myntra, you must have noticed this thing, that let us suppose only if you are like ordering one kajal. So the kajal, one pack of kajal is being like delivered in one box, and the box is having so much of the paper cutting and only a small patla sa kajal. So it is specifying what? It is specifying the package is so big, product is so small. In the same way, 99% of your DNA is junk DNA, and this junk DNA is protecting your gene. Gene means genetic material, and we are trying to study this DNA because we want to study the information about the gene. Let us suppose I want to study about the DNA and I am taking up this part as gene. Now you would be wondering, ma'am, how we are trying to distinguish that which one is junk, which one is important and which one is gene? Now, try to understand here. You can distinguish in terms of the gene and DNA when you have the knowledge of the nitrogen base. And these nitrogen base are nothing. They are the structure which are considering some part of the gene or the genetic material. How? We'll try to understand. If everything is clear to you, then let me proceed further. Yes or no? Yes? Okay, fine. So let's try to revise it again. So what we had done, we had taken one cell. From the cell, we had considered the nucleus. Nucleus is being zoomed out, and then we are taking a what? A shape. So the shape is seems to be like this. So it is like the nucleus is being filled up with numerous of the chromosome. So human body, it includes what? It includes 23 pair of chromosomes. Okay, so this 23 pair of chromosome, it means 46 in number. So your cell is being filled up with 46 chromosome. Now, your cell, they are like one cell is including what? 46 chromosome. Fine. So this is done. Now, your body, let us suppose if I am taking one cell out of it, let us suppose I am taking another cell from, out of it. So let us suppose the cell from this left hand is having 46 chromosome, cell from the right hand is having 46 chromosome. Let us suppose I am taking one cell from the forehead, one cell from the cheek, one cell from the hair, and in this way every single cell is having 46 number of chromosome, 46 in total number. But consider one thing, every single cell and their chromosomes in terms of number as well as their DNA and genetic material is going to be same or similar. I mean, if I would be taking cell from here and zooming it up and taking up the DNA, so the genetic material of this DNA and the genetic material from the cell of this hand is going to be same. It means it doesn't matter. I am getting the DNA material from the blood, or I am taking the DNA material from the hair, from the skin, from the nails, from any tissue. The genetic material is going to be similar. Fine? Similar for one individual. I mean, my DNA, my genetic material is going to be same. Even if I am taking it from any cell, from the hair, from the nails, from the skin, from the even further when I would be teaching you, you would be wonder that yes, saliva can also provide the DNA material. Fine. So there are some category of secretor and non-secretor. So that's why. Fine. Okay, roughly tell us about the, okay, fine, I'm telling you. Fine, so it means they are 46 in terms of number. Now here, when I am taking up one chromosome and trying to study about this chromosome, so I find out that one chromosome is made up of two chromatid. This side chromatid, this side chromatid. So chromatid is nothing, they are, let us suppose if this is the DNA, so this shape and this shape, they are nothing but they are taking up one chromosome. Now this chromosome is being further stretching out and the most condensed part is chromatid, then the less condensed part is chromatid, and when I am stretching it further, this part, this double stranded structure or double helix structure is nothing but it is the DNA. DNA is the deoxyribose nucleic acid and it is being considered as what? It is being considered as the gene. I hope now you have understood the basic parts. That's how we used to specify about the gene or the genetic material. Now, just to give you the basic overview so that further you can understand the other things related to the class, I need to explain more about the DNA. or the basics about the DNA, like what do we mean by the word DNA, how it is different, and how this chain is being maintained. Okay, so what I am doing here is I am cutting this part. So I am cutting this part, taking it up to the next slide, and we'll try to understand. Okay. So it is nothing but it is a double helix structure or double stranded system. So when I'm trying to study this part, so when I am trying to study this part, okay, so when I am trying to study about this part, first thing I can understand that there is something in terms of a bond which is joining together this structure. I mean, here I can understand that there is one stream, like here. Now you would be able to understand. Now try to understand. Okay, so one strand you can see in red color, another strand you can see in orange color. Okay, so this is what, this is DNA. Now the orange color strand and the red color strand, they are attached together with one horizontal line, as you can see, and this horizontal line is binding them together, and this binding is very strong. Now here we need to understand what is this bond and how this DNA and this chain is being specified, how this chain is being structured together. Okay, now let's try to understand. One more thing, in today's class I am just giving you a brief about it in a detailed manner. What do we mean by DNA structure? This I would be taking up in the unit 1.7. Okay, so unit 1.7 is going to be somehow specific one, so wait for that as well. Okay, now when I'm trying to understand this red strand and this orange strand, I can understand this phenomena that these bonds, they are something which are strong. So when I try to understand about it, I got to know about the nitrogen base. Means in the human body, there are something called as the nitrogen base, and these nitrogen base, they are specifying about what they are specifying about the double helix structure. Okay, so what do we mean by this double helix structure? Let's try to understand. Now, nitrogen base, they are being specified by the name NB. So normally a human being is having 5 type of nitrogen base. So we are having 5 types of nitrogen base. In the body, that is A, T, G, C, U. Okay, so A, adenine, thymine, guanine, cytosine, and uracil. Okay, so these 5, they are the nitrogen base. Now here bond is being created. So bond means the bond must be between the 4 person, and the 5th one is going to be the extra one. So the same is the case. In case of DNA, to create a bond, we require the 4. So therefore, we are considering A is making a double bond with T, G is making a triple bond with C. This is the basic structure that we have. Okay, now the question arises how this chain is being maintained. Now we'll try to understand how this chain that is being specified and how this chain is being maintained. So before that, I just want you have you framed this structure in your notes? Yes or no? So I am just giving you 30 second ka time so that you can frame it in your notes. Done? Okay. Write down this also. Done with this? Yes? Okay. Now, we are having a proper structure. So, let me frame that here. What this structure is going to define. Okay. So, first, there is going to be sugar. So, sugar and nitrogen base, they are going to bind together. Fine. So, just wait for a while, just write down. First, sugar and nitrogen base, they will bind together and they will create nucleoside. Now, the nucleoside that we have prepared now plus phosphate will bind together and they will create nucleotide. Now, nucleotide will create a chain. And this chain is nothing but called as what? DNA. And this is what you need to remember. I will repeat it multiple times so that it would be stuck in your memory. So what I am saying, first what we would be doing is we would be taking up the sugar. Sugar and the nitrogen base, they are bind together and it will form what? The nucleoside. Now the nucleoside would bind together with the phosphate that's there in the body, and with the formation of the nucleoside and the phosphate we would be having nucleotide. Now a chain of nucleotide, it will be creating what? It would be creating the DNA. Okay, now we'll try to know about it in a great detail and depth, and we'll try to know what is the role of the sugar, why we are using the sugar, and how with the help of the sugar we are creating it. I hope you must have written this. Okay, so DNA structure is considered as a kind of structure which include some sort of like carbon base. So DNA structure is having 5 carbon sugar, and that carbon sugar is nothing but it is deoxyribose sugar. Fine. So let me write down here. So sugar deoxyribose sugar. For what? For DNA. Okay. Now DNA structure is nothing but it is having a 5 carbon sugar structure. Now this 5 carbon sugar structure is called as deoxyribose. This sugar is considered as the essential one, and the reason why, because it is being considered as the backbone of the DNA strand. Fine. So let me give you the basic overview, or we can write down here just to understand about it, the role of sugar. Okay, so what is the role of sugar when we are trying to create the DNA structure? So first, so try to understand. So let us suppose I am saying A. So let us suppose A is like this. Then I am saying T. So T is like this. And let us suppose I am saying G. So G is like this. And then C. C is like this. In the DNA, we are not having uracil. In the DNA, we are having A creating bond with T, G creating bond with C. Okay? So this is the basic structure we have. Why we are not having uracil in the DNA, I will let you know, but wait for that. Here only try to understand this way. Now, DNA is nothing but it is a chain. First, what we are doing? These are what? These are nothing but these are the nitrogen base. Okay. Scientifically, how it would happen, I will let you know in the unit 1.7. Specifically, okay, here I am giving you general view because I know there are so many students who are not from biology background. So because of that, I am taking this lecture as very generic. Now in the unit 1.7, I will be taking up you into the next level. Okay, so just wait for that. That's why I took this lecture as the first and the introductory lecture for the physical anthropology, because in the unit 1.7 we have to be very much specific. But after unit 9.4, I would be taking up the lecture 1.7. So just wait for that as well. Okay, now there is sugar. Let me form the sugar in this format. So try to understand. This is the sugar. Now what I would be doing? I would be taking up sugar and nitrogen base. So sugar and nitrogen base, they would be creating nucleoside. The nucleoside, they would attach with the phosphate and it would form what? The nucleotide. Then there is going to be a chain of nucleotide. Now you know this thing very well. Just tell me, A is going to create bond with whom? So A is going to create bond with whom? And G is going to create bond with whom? So answer me quickly. Normal. And quickly, tell me the answer. A is going to create bond with whom? A with, okay, Swati is saying T. Okay, G with, G with C. Very good. So, sugar is very much important. Why? Because Because sugar is become the pioneer for creating the chain of the DNA, the chain of the nucleotide, and this chain of the nucleotide would form up what? The DNA. Now you had seen that DNA is this structure. We'll try to understand how this structure is formed. So first try to understand, whenever we are creating the DNA, so let us suppose this structure is being formed. Could you understand? This is showing what? This is showing A this side and T this side. Now I am using the color so that you can understand. Okay. Now another thing. G is creating bond with C. Okay. Now you have understood one thing. Let me form it further so that you will get the basic detail. Here, based upon the color coding, you will get the basic idea that these DNAs, they are stuck together. Okay, now based upon the color coding, you can understand that whenever A is here, for sure T is going to come and bind it. Then whenever G is there, C is going to come and bind it. Here, based upon this way, you can understand that one nitrogen base is having three strand. Three strand means what? 1, 2, 3. So one nitrogen base is having three strand, three sides. Each side is binding with someone. Now here, take up this A. Take up this A. Let me take up the different color pen. So let us take up this A. This A from upside. This A from upside having bond with whom? With C. This yellow color. And this A from the lower side having bond with whom? With G. And this A on another side is having bond with whom? With T. It means they are binding together each side. And this thing is being inculcated by whom? This is being inculcated by the sugar. So this nitrogen base is not binding. It is the sugar which is binding this nitrogen base with the another nitrogen base. So from this perspective, you can understand that one single sugar connects to, so it is being formed in this way that one sugar connects to one phosphate group it is connected one with one nitrogen base and it is being connected with the phosphate of another nitrogen base. So this is how it is being formed. And this is nothing but the usage of the sugar. Now another important sugar used to provide, that sugar provides stability. So I am erasing it to DNA structure. Another thing, sugar provides support to DNA structure. So it is providing some sort of support to the DNA structure. Fine. So that's how this is the basic usage of the sugar as a role in the part of the DNA. And this is how I hope you have understood. So it means my DNA is being formed, and this DNA, like this is how it is being formed. One more thing, try to understand. A is creating a double bond with T, G is creating triple bond with C. But the line are so thin, we are not, like these are not visible. Therefore here, just for the reference, we are only showing a single line. Instead of that, we can also show like this way. Let me give you one overview. Like in this way, I can show like double line and here triple line. So in this way, we can also form the structure triple line, then double line, then here triple line. Okay, so that's how this structure would look like. But if you will see far away, like from the far away, you can see here that these structure, they are being specified in a single line, in a single bond. Fine. So these are, that's how these bonds are being specified. Fine. One more thing here, try to understand. There are multiple type of bond. So there is peptide bond, there is hydrogen bond, there are different type of diester bond, ester bond. So there are different type of bond. Again, I would say here in this lecture, I am just giving you a basic reference. What are the different type of bond, how these bond are being specified. This is what I will let you know in the unit 1.7. Fine. So first we would be taking up this unit 9.4. After the completion of unit 9.4, we would be taking up unit 1.7. So just wait for that. That unit 1.7 is going to be very much specific. So you would be thinking that, you would be amazed that how in our body the God has beautifully created the structure. Each and every part of our body, they know their work, they know their usage, they know their function, and beautifully they are being created in terms of a structure that one thing is attached with the another, and they are working phenomenally. Fine. Okay, so from this perspective, you have understood the role of the sugar, and here you have understood one more thing, that based upon this three side bond, every single nitrogen base is attached with the another nitrogen base, and that's how this structure is being completed. Once it's being completed, it is being bind together. So it is being bind together, and based upon this binding, we can basically understand that how, like based upon this binding, we can understand the structure. Can I get one wire or something? Okay, so just try to understand here, like let us suppose, it seems to be very small, but let us suppose if I am winding it, so it would be like, hope is it visible to you? Like, so it is being in terms of a DNA structure, and this structure is here you can see, this structure is here you can see is being specified or similar to this one. Fine. So when I will trying to wind it up, so if I am winding it up, so because of, if I will wind it, it will create a DNA structure. If I will unwind it, so the unwinding would be seems like this. It means it is the DNA only. These DNA, they are being winding up, and how they are being winding up. So there is one protein called as histone protein. Here in this structure, you need to understand that these winding up, here you can understand that these winding up, they are together attached with one histone protein. So DNA is attached with this histone protein. Then there are so many histone protein, and DNA is attached with it. Fine. So because it is attached, so therefore it is coiled together. It's coiled like this. So this is winding. If I will unwind it, So when I will unwind it, I will get two straight lines which are bind together. So these straight lines, so see, this is the zigzag manner. This one is again winding up of the strands. And now if I will unwind this strand, I will get this structure, and this structure is being specified as the DNA structure. Fine. So have you understood till now? Just write down about it. Done? Okay. Now, till now we have understood that sugar is creating bond with the nitrogen base, then nitrogen base and sugar creating the nucleoside. How would you remember? S for sugar, S for side. So when there is sugar plus nitrogen base, it would be nucleoside. Then nucleoside and phosphate. Phosphate T sounds much, so therefore it would be creating nucleotide. Then there is a chain of nucleotide. So the chain of nucleotide would be specified as what? The DNA. Okay. Now, we said that there are 5 nitrogen base, but for the creation of the DNA, we require only the 4. So therefore, we are doing some sort of discrimination with the uracil. It seems to be, but this is not the case. There are some certain reasons, there are some certain justification why uracil is not part of the DNA. Let's try to understand. Why uracil not part of DNA? Okay, so this is what we have to do. The first thing is this: G is creating bond with C, and that's also triple bond. Now A would be there, adenine would be there. Now A have to choose between the T and the U. We are having the genetic material, and for the genetic material we are having DNA. Another part, as in terms of the part of the protein synthesis and the protein translation, we are having RNA. So DNA, deoxyribose nucleic acid, RNA, ribose nucleic acid. Now RNA, basically the ribose nucleic acid, it is having the bond of what? A with the U. So they are having uracil. It means it's A who's going to create bond with T and with U. When A will create a bond with T, it would be the DNA, and when will create a bond with U, it would be the RNA. So herefore A is the one who is the deciding factor with whom A is going to create the bond. So A is creating bond with T, means preferring the T over U for creation of the DNA. The reason behind that is T, basically the thymine, is being considered as a phenomena, a nitrogen base which is there for creating this strong stability. So the T, basically the thymine, is specifying as a storage, like a storehouse of the genetic information, and that too for a long period of time. Fine. So here why we are preferring, so thymine is more stable than uracil. So thymine is considered as more stable than uracil, and it is chemically more resistant to damage. Thymine is chemically more resistant to damage. Now it's difficult to damage the thymine. We like comparatively it's difficult to damage the thymine. It means our DNA is going to be persistent and it's difficult to damage the DNA. Now try to understand further when I would be teaching you other aspect, and even in this lecture also when we would be talking about the aberration, you will get to know that even a minute change in the DNA structure can change the life of a person. So these error, they can somehow maybe creating one person as phenotypically male but internally as a female. Means from your internal structure you are a female but phenotype, your body structure is like a male. So this type of changes. Another is, so there are some sort of genetic error in terms of some are having the polydactyl, means we are having the 5 fingers, so some may be having 6 fingers. So you must have seen the there is one famous Bollywood celebrity Ritik Roshan. So he is having six. He is having two thumbs. You must have seen. Okay, so not only this, some person they used to have the mental retardness. Okay, so this type of things they used to be there. Therefore, we want someone to be stable and someone who's resistant to the damage. So this is one of the reason for that. Another reason that why we are not preferring uracil is in terms of the error detection. Fine. So what happened exactly is cytosine. Cytosine means the C. So as you know, C is creating bond with G. So there is no such debate. C is going to create bond with whom? But C used to change, and sometimes spontaneously C used to change into U. So C spontaneously it used to change into this uracil, and there is one chemical reaction. The name for that chemical reaction is de-deamination. So there is one chemical reaction. So in this chemical reaction, C used to change into the U. Now it would be difficult. Let us suppose C is undergoing the change and there is any chemical reaction, and because of this chemical reaction C is changing into the U. Now there is the nitrogen base, original nitrogen base U present in the body. There is chemically changed U is also present in the body, that is being changed from C to U. Now it would be difficult for A, if A is creating a bond with U, to determine which U is the original and which U is the changed form of C. And if in any case A is creating bond with the changed form of U, that bond is going to be error based and it's going to be difficult to differentiate and to provide stability to the DNA material. Therefore, we are just skipping this error to happen and we are taking up T-thymine, which is also the stronger one as well as the stable one. So that's why by using it, so just to avoid it, we are having this phenomena. Okay. Hari saying, which will consider in using the investigation on crime and medical purpose. Are you talking about the DNA and RNA? Are you asking for that? Yes or no? Yes. Okay. So it's DNA only. Okay. So just wait for that. Further, I'm going to differentiate between the DNA and RNA. You will get everything clear in your mind. Just now try to grab the new things as soon as I'm telling you. Okay. So here Again, I am repeating, there is one process, a chemical reaction, diminution. In this chemical reaction process, C, the cytosine used to change its form in the uracil. Now, original uracil is also available in the body and changed form of uracil is also there. Now, based upon this process, A would be it would be difficult for the A to differentiate. Therefore, A is preferring to create bond with T. Fine. So that's why T thymine is providing stability. So here, write it down. To avoid error in choosing the original uracil and changed form of uracil, okay, to avoid the error in choosing the original uracil and changed form of uracil. That's why we are choosing what? We are choosing the thymine. Okay, so that's also one of the part. Another part is in terms of the functional differences, like DNA carrying the genetic material is permanent, it is strong, whereas uracil is not that much permanent. So therefore, RNA is using the uracil, and because RNA is using the uracil, therefore, in terms of the RNA, we are basically in terms of the RNA, we are only taking up the temporary information. So there are different types of usage among the DNA and RNA. I will let you know the usage as well. Further on, when we would be taking up the DNA replication and that process. Fine. So just consider this as a part of as a part of the process. Fine. So let me give you a basic quick review about everything we have done. First, we had taken up the cell, then out of cell, the nucleus. From the nucleus, chromosome. Now, zooming it out, chromosome. From the chromosome, chromatid, then chromatin, then DNA. Now, trying to understand how the DNA is formed. So, taking up nitrogen base, then sugar, then nucleoside, then phosphate add-on, then nucleotide, then there is a chain of nucleotide. So we are having 5 nitrogen base, A, T, G, C, U. Now not taking up the U, we are creating A double bond with T, G triple bond with C, and because of these bonds we are creating up what? The DNA structure. So we have understood the basics of the DNA structure. The DNA structure is not as much simple as I taught you. It is much more complex, and we'll understand that as well in the unit 1.7 again. Okay, you would be saying, ma'am, you have repeated the 1.7 again and again, but there are so many new students they are taking up, would be considering, ma'am, it's so generic. But today deliberately I am making this class generic so that you will get the basic idea. Once you will get the basic idea, then I will dive deep and then will let you the specific details. Okay. So till now, if you have any doubt, you can ask for. So I am waiting for 1 minute and then we would be starting up further. Done? Okay. Now, we are trying to understand about what? We are trying to understand about the chromosome. Now, let's try to know some basic aspect about the chromosome. We have understood that chromosome is nothing but in terms of a structure, like we have seen the internal structure, but let's try to naming out the external structure of the chromosome. Okay, so for the study perspective, as I told you, I would be taking up the DNA in this format. Fine. So this is something specified in terms of the DNA. Now DNA is having like the DNA structure in this format, like I can take up this R or can take up this R. Fine. The DNA is going to be same or similar. As I told you that one chromosome is having the 2 chromatid and one pair of chromosome is having the 4 chromatid in the same way. One chromatid is having one DNA. Even if I am stretching the DNA from this side or if I am stretching it from this side, still these two are called as what? Sister chromatid. Fine. So this one chromatid, let us suppose A. This one second chromatid, let us suppose B. So chromatid A is having one DNA. Even if I am stretching it from here or being stretching it from there, still this would be considered as one DNA. Now these two are, they are specified as what? Sister chromatid. So these are what? Sister chromatid. Okay. Now we are having one pair of chromosome. One pair of chromosome would be having like two sister chromatid. This one sister chromatid, another chromosome. These are also sister chromatids. Then again, so one pair of chromosome is having two sister chromatids. I mean two pair of sister chromatids. Fine. And one chromatid, one DNA. If stretching it from here, same. If stretching it from there, same. It's going to be same or similar. Fine. Okay. Another thing. It means there is no arm and legs. Both sides are same. If I'm standing here, taking up this arm, same DNA. Let us suppose if I'm standing there, upside, and I'm stretching it from there, this side, so out of the board, again DNA is going to be same. Therefore, these both are called as arm. They are not called as legs. So we won't specify these are arms, these are legs. Why? Because I'm standing like this. No. The DNA structure and the basic phenomena regarding the DNA structure is same. Yes, Navya, this foundation course, it also includes the, okay, so this foundation course is for anthropology. So it's Anthropology Optional Foundation Course. Yes, all the points in Anthropology Optional syllabus, every single point. Fine. So here, these are what? These are arms. Now, it would be difficult for me if I would be calling both as arms. So it would be difficult just to give the position. Let us suppose if I'm standing like this, I need to specify upside arms and downside arms. So just to specify that, we used to provide some name. And the naming is being specified in this way. This as P arm and this as Q arm. It means the lower side arm is called as Q arm, upper side arm is called as P arm. Fine. Now another thing, the end of the P arm or Q arm is called as what? Telomere. Okay. So end of the arm be it the P arm or be it the Q arm. So this end or this end, both would be called as what? Telomere. Now, center is called as what? Centromere. This is what we are naming the shape of the chromosome. So, the center is specified as what? The centromere. Fine. Now, in some of the chromosome, there is extra, like this is something called as normal. Now I am making up another one. I hope you have written it there in your notes. Yes. Now let me give you the overview again here. So normally there is going to be this. Normally there is going to be this structure only. Okay. But sometimes it happens like this is centromere. Like this is centromere and this is telomere. And this one PR and this one QR. Fine. So now this PM and QR, sometimes it happens that the satellites, like these chromosomes, they are not ending at the point of telomere. Like for example, end of the arm is called as what? Telomere. Fine. Sometimes it happens that the end of the telomere is not ending the chromosome. It's having further material attached together, and this is in this format. This is there in this format. Okay. Now, we had specified that end of one arm would be called as telomere. If this is the case, look, this is the normal case, but if there is extra chromosome being attached together, if this is the case, then again end of the chromosome would be specified as telomere and would be called as telomere. Normally, this should be called as telomere, but now this is not the telomere. Why? Because end is this. So therefore, this point is being specified as secondary constriction. And this part as primary constriction. Okay. So normally this midpoint would be specified as centromere, but if there is extra material being attached, and this part would be like these parts, they would be specified as satellite chromosome. These are specified as what? Satellite chromosomes. Fine. Okay, exactly. The extension form of the DNA, this would be called as what? Satellite chromosome. And this is how, in terms of a structure, we used to define. I hope you can understand. Fine. Now, let's try to know some basic fact about the chromosome in terms of basic structure, and then we would be taking up our topic for today's class as in terms of numerical aberration. Okay, I hope you have understood till now. If yes, then well and good. Let me proceed further. Now, As I told you that human being, they are having 23 pair of chromosome. It means 46 in terms of number. Now, these chromosomes, they are in terms of pair and they are specified, they are arranged in a serial number. And how that serial number is being specified? Here, we are having this structure. Okay, so this structure is being specified. Fine. Now here, this is the 23 pair of chromosomes are attached, they are arranged. You can understand that there is some variation in terms of shape. Let me tell you, the first chromosome, is the biggest one, largest one in terms of shape. And the 23rd chromosome, it is the smallest one. So as soon as we are going further from the first chromosome to the 23rd chromosome, we can specify the basic shape. Here also, you can understand that this shape is being arranged. One part is including 23 chromosome, another part is also including 23 chromosome. This is here I am giving you in terms of variation. Like 23rd pair of chromosome is called as sex chromosome. And 1 to 22nd, they are called as autosome chromosome. They are specified as autosome chromosome. Now in terms of the Navya is saying that ma'am, can we specify primary construction as centromere? Here try to understand, primary construction is centromere only, but because there is satellite chromosome, therefore we cannot say it is the center. Reason is this. Now look, earlier your P arm is this, but now your P arm is this. So how it could be center? Because P arm is the bigger one, the Q arm is the smaller one, and this one is attached further. Therefore, we are giving it another name to just specify. Otherwise, it is center only, primary constriction. Okay, so kindly avoid using the name centromere in case of satellite chromosome, but call the centromere in the satellite chromosome is primary constriction. Fine? Okay, now 1 to 22nd, these are called as autosome. Autosome chromosomes, they are important one just to frame the body structure. Like how would be my nose, what would be my skin color, how would be the shape of my eyes, how the face structure is going to be, how the hands, the basic part of the body. These are being specified by the autosome chromosome. But what would be the gender of the person and what would be the hormone related to that gender is specified by the 23rd pair of chromosome, that is the sex chromosome. So the bodily structure, the phenotype between the male and female is being distinguished and being determined by whom? By the 23rd pair of chromosome. Otherwise, every single human being, they are having two eyes, they are having one nose, they are having 2 ears. In terms of structure, it is completely similar. Only difference between male and female, it is being there due to this 23rd pair of chromosome. Now, every single human being, they seems to be more or less different. But you must have seen that siblings used to be similar. We used to we used to get the idea that these two persons, they are related because their their features, their features of their face, they seems to be the same, seems to be the similar one. So that's how we used to specify. So from this perspective, you need to understand this perspective, that because in the siblings there is matching in their DNA. So let me tell you, among the siblings 74.5% of their DNA is similar. Therefore their autosomes used to be similar, and that's why they used to look alike. Their nose, their eyes, their face features, they used to be same or similar. Okay, fine. Now these part, they are called as what? Ploid. Means this orange color table, it is specified as what? Ploid. Fine. So we are having what? We are having, like, one ploid is called as N. So we are having two ploid. So two ploid means diploid. Because we are having 2n. Now, when we are taking up half the number of ploid, they, like human being, they are having what? Haploid. So let us suppose you are having diploid. So half the number of ploid. How many ploid we have? We have two ploid. So half the number of ploid, we are having haploid. Fine. So because we are having 46 number of chromosome, therefore we are having a diploid. And when we are taking up haploid, means half the number of ploid, so the half the number of ploid would be specified as the ploid only. So half the number of ploid would be specified as ploid only. Okay. So this is the basic structure, and this is done. Now you have the basic idea. Now I can take you towards the numerical aberration. Done? Okay. So now let's try to understand something about the chromosome, and further on we'll be explaining the basic about the structural as well as the numerical aberration, like how structurally the shape would be different, how numerically the number in terms of the total 46 can change it. Hope you understood everything about it. In terms of the PPT, like as I told you that you need to subscribe the official channel of the Sunya IAS. So in the official channel of the Sunya IAS, that is basically Sunya IAS Anthropology. So in that Telegram channel, I am providing these PPTs by naming with the lecture number and the topic name. From there you can get these PPTs in terms of a PDF. Okay. Now let's explain about the chromosome and then about the error in the chromosome. So the word chromosome, it comes from the Greek word chroma. Chroma, it means color, and chromosome, zome means body. So chromosomes, they become visible when stained under a microscope. Means chromosome, we won't be able to see the chromosome. Chromosome we can see only when we would be putting some color. Where we will get more and more stain, there we would be specifying more chromosome or condensed chromosome is there. Fine. Now chromosomes, they are what? They are tightly packed, and you know very well that the most condensed form of chromosome is called as what? Chromatid. And the tightly packed form of DNA, they are found in the nucleus cell. It is visible in the nucleus cell, and they contain both gene which carry some instruction for the traits, as well as it includes some non-coding region as well. Non-coding means the part which is not coding for anything. What is that? How it is being specified? Now let me give you the basic overview about it. As I give you the basic idea about how these DNA, they are framed together. Okay, so now let's try to understand. Let us suppose this is, what this is the A, and let us suppose this is the B. Sorry, this is the T. Now this is the G, and now which color to take? Let it be green, and this is the C. So from this perspective, you can understand that these are being bind together with each other. Okay, as I told you, let us suppose if I am taking this DNA, let us suppose that this is this DNA. Here, could you see? Hope it is visible to you. So this is the DNA, and this is how I am taking this DNA. In this DNA, these parts, they are attached together. They are attached so condensedly that I can't see the bond in between. But let's suppose I started unwinding it. I can see some space in between. Let us suppose I will unwind further. If I will unwind it further, what I can have? Let us suppose I am unwinding it. So what I can have? I can have this structure. Can you understand? I can have this structure. Now, in this structure again, let us suppose have the same thing. I am taking this part. Okay, hope I am taking this part. Now, here you can easily understand. Something is creating bond. Let us suppose this is A, this is T, this is G, and this is C. So they are creating bond. It means for the purpose of the study, I can take up this thing, and based upon this, we can study. Fine. I hope you have understood this thing. Now, sometime it happens that because of the process of the DNA manufacturing called as the DNA replication process, some part of the DNA they used to hide themselves, or sometime they used to get separated out of the chain and they used to miss. And because of this, sometime it happens that because of this we used to find out some sort of error. And that's how it is giving birth to the concept of chromosomal aberration. So chromosomal aberration is nothing. It is made up of two words. Chromosome means something specifying about the genetic material. Aberration is nothing but it is in terms of error. When there is some sort of error in the genetic material, these error they are being specified, or these error they used to occur in terms of sometime in number of chromosomes, sometime in the shape of chromosome. Shape means the shape is going to be different. Fine. So that's how we used to take up the chromosome aberration. Now, the chromosome aberration, it could be specified into chromosomal aberration. Okay, so we are taking up the chromosomal aberration. So chromosomal aberration, primarily they are of two types. How these two types are being specified? So the first one is structural, another one is numerical. Okay, now structural aberrations, they are being specified into two parts. I mean, sometimes in the structure there is changes in terms of the gene composition, sometimes in terms of the gene arrangement. Sometime in the composition, so gene composition, sometime in the gene arrangement. Okay, then in terms of the gene composition, we used to have two forms out of it. One, when there is some sort of deletion, and when there is duplication. In terms of the gene arrangement, there is inversion and there is translocation. There is translocation. Now, in terms of the numerical aberration, there is euploid. Ploid, you know very well that this one part is specified as ploid. Fine. So for this, we are having euploid as well as we are having nuploid. Another one like we have in terms of nuploid. Now euploid and nuploid, they are being specified in terms of trisomy, monosomy, triploid and tetraploid. Okay, so nuploid. Tetraploid, monosomy and trisomy. So while taking up the time into consideration, we won't be able to complete numerical chromosomal aberration in today's class. So therefore in today's class we would be taking up the structural chromosome into consideration. Fine. So let's start the structural chromosomal aberration and let's explain about it. Okay. So for the structural chromosomal aberration, we are having first one as being specified in terms of deletion. So I won't be repeating it again and again. You know it very well that first we are taking up one DNA that's in the winded form. We are first unwinding it up. After the unwinding it, we are getting a double-stranded structure. Now this double-stranded structure is attached with each other. So now try to understand. I am having my DNA structure here. So here is one pair of chromosome. Okay, this is something that you know. This is what the pair of chromosome. Now this pair of chromosome, let me take it one by one so that you would be able to understand. Okay, fine. So this is the DNA. First, it is being stressed in such a way. Fine. Let us suppose first it would happen. Now, what I am doing is I am taking it further and what I am doing is I am unwinding it up. Hope you have understood. I was having this structure. After unwinding it up, I will get this structure. Okay. Now, let me name it 1 and let me name it 2. You have understood there is one chromosome. From this one chromosome, I had taken these two arms. If these two arms, they are unwind together, I will get this horizontal structure. Let us suppose if I am cutting it from here, let us suppose cutting it from here and taking this form. So now assume I am taking 1 here and 2 here. Can you understand? It's a step-by-step phenomena. What I did, first I took one chromosome, I took the DNA part by stretching it, then I it up and I got these horizontal DNAs, and you have understood the horizontal DNA, how I will get the horizontal DNA. After that, I am cutting these and placing it like this so that you can understand. Now I just want to name it. Kindly do not think here I am giving name to the nitrogen base. No, I just want to simplify the explanation, and just to simplify the explanation, I am naming them like one square. I am naming one square only so that I can tell you the change. Therefore, I am naming one square. Let us suppose it is A, this is B, C, D, E, F, G, H, I, J. And this is small a, B, C, D, E, F, G, H, I, J. Fine. So this is what I am naming. I hope you can understand. Yes or no? Okay. Now, what happened is this is nothing but this is the normal chromosome. Now, in the process of replication or in another process, what happened is there is some part which is deleted. So there is some part which is deleted. Because some part is deleted, we used to specify that some part out of it, it's being deleted. This again going to be same. So A, B, C, D, E, F, G, H, I, J. So this is first one. Now what happened is in the process there is some part which got missed. So A, B, C, D, E, F, G. So let us suppose H, I and J means this part. This got missed. So therefore this would be called as what? Aberrated chromosome. It would be specified as aberrated chromosome. Now this was one. One means this. This was 2 means this. In the process, when we were having the process, some part of these 2 got missed. If it got missed, now taking it up, this is the normal chromosome. But because some part is deleted, So therefore, and deleted from where? From 2. So therefore, the shape for 2 would be like this. It won't be completed. It would look like in terms of error. So therefore, this is aberrated chromosome. Means error happens, and because of that, the shape of the chromosome affected. I hope you have understood what would happen in case of DNA deletion. Hope you can understand. If you have any sort of difficulty anywhere, let me know. Could you understand? Do you have any sort of doubt? If yes, then let me know. If you have doubt at any point. Now we have understood deletion. Now let's try to understand the duplication. So duplication is also being specified by the name where where we used to specify addition. I mean, there is going to be some addition. Something used to be added upon. Fine. So this one is deletion. Let me specify the second one. Second one, addition or duplication. Now, I'm not going to do the same. I will just taking up this only so that you can just understand. Okay, because you have understood how it would happen. I'm just giving it name. So, how this will happen. So, in case of addition, A, B, C, D, E. Let us suppose we are also having F. This is from 1. A, B, C, D, E, F, and let us suppose it is from 2. Now this is the normal chromosome. Now because aberration would happen, so how it would happen? A, B, C, D, E, F, and then we are having A, B, C, D, E, F, and because there is going to be duplication or addition, let us suppose E, F again added upon. So this is 1 and this is 2. So your normal chromosome would look like this. And after the aberration, because in the arm 1 everything is okay, but in the arm 2 something is added upon. So this is aberrated chromosome. So there is aberrated chromosome. So aberrated chromosome again changing the shape. Deletion chromosome again changing the shape, but then we have inversion as well as translocation. Fine. So how it would happen in case of inversion and translocation? Let's try to find out. So the third one is inversion. As the name suggests, inverted. Fine. So let us suppose again we are having A, B, C, D, E and F. And we are having A, B, C, D, E and F. So this one is normal. When it's being changed, so one is going to be the same So the changes would be here. EF was there, but it's inverted. In the process, EF, this was the series, but it's inverted. FE would be there, means inverted. So this would be the next. Now they were having 6, they were also having 6. That's why we can understand that in terms of the shape, for the normal and as well as for the abbreviated, shape is going to be same. Shape is going to be same. Even in case of abbreviated, shape is going to be same. And that's how it is being formed. Hope you can understand. So the basic pattern was EF, but it changes into the FE. So it is inverted. So the shape would remain the same, but it's inverted. Fine. Now the fourth and the last one is translocation. How the translocation is being formed? It is specifying 1 and A, B, C, D, E, F specifying 2. Now this one is normal. Now it's being changed. A, B, C, D, E and F first. Now what would happen? A, B, C, D. Now it is E and F would be located here, and capital E and capital F would be located here. So this one is abbreviated. So it means B2 is translocated at one place and 1 is being translocated at another place. So there could be small e and small f. So this one is abbreviated part and this one is also abbreviated part. So again here also in terms of shape, your DNA or chromosome would be the same. This one is the normal and this one is the abbreviated. Okay, so that's how it is being specified. Okay, so Hari is asking, is the structure same in deletion and aberration? What is the change between deletion and aberration? It seems to be similar. I can't understand your question, Hari. Aberration means error. Deletion is one of the type of aberration. Aberration, they are basically of 4 types. Aberration, they are basically of 4 types. So in the aberrated chromosome, one part is being missed. Kindly reframe your question and then ask me again, because we are done with the topic. I am just waiting for your doubt. Which seems to be similar. Okay, so basically aberration means error. Error could be of any type. In case of deletion, some part of one arm is missed out. So in case of deletion, one part is deleted. not the complete DNA, but one part from one arm is deleted. So here, any sort of change I am marking with the red pen. So we are having here Hij, but here Hij, they are not there. It means they are deleted. In the process of the DNA replication and otherwise, this part is being deleted. Now, in case of addition, one part is added and this part is being like some sort of duplication. So, in case of addition, EF, this part is being added 2 times, means duplication. It means duplication. So based upon this, we can say that the aberrated, like this one is small, this one seems to be bigger one. So this part is being aberrated. Fine. Navya is asking, ma'am, if deletion occur, is there any chance to delete whole strand of one side? No, not at all. There is no chance that the complete strand is going to missed out. No. Why? Because there is a strong bond. Quite possible one strand or two strand they may missed out, but complete strand cannot be missed out. Fine. So this is what? This is the duplication. Now, in case of inversion, inversion means what? Inversion means EF. This should be the series, but the series is being changed. It is being inverted, and that's how we used to specify that this part is being inverted, and from this perspective we can say the shape would be the same, but there is in terms of error in terms of structure. Now another part is translocation. So the translocation is in terms of that two arms, they are mixed up together. So two arms, some part of two arms are mixed up together. Okay, so that's how it used to happen, and this is the basic phenomena we used to have. I hope you got the clarity. If yes, then let me know. Fine. Okay, then well and good. So we are basically done with the first part, and the next part we would be taking up in the next lecture. Next lecture we are going to have on the Friday. Fine. So like next Wednesday and Thursday we won't be having the lecture. So on the Friday we would be having the next lecture where we would be taking up the next part, and that's going to be very interesting one. It's not so much technical. The next part is not that much technical. It is something which is informative, and we will be doing that in terms of a very like exciting or very interesting way. So stay tuned for that. I would be notify you through the Telegram channel, and this class PPT I would be providing there only. Fine. Now let me just tell you that we are Navya is asking, ma'am, does two strand join? Okay, so it is strand. Okay, so she is asking, ma'am, does two strand join anywhere in any case? Sometime it happens. What could be those times? I will let you know. But that would be part of the unit 1.7. I mean, when it would happen, like one part would be cut from here and there and can be added upon the other part. Like there are very rare to rare chance that translocation would happen. Usually it's not there. Usually what happen is usually deletion used to be there. Moreover, and sometimes addition could be there. Inversion and translocation, they are very rare phenomena. Okay, fine. So as I was telling you that we are starting our new batch for the Anthropology Foundation course for covering the complete syllabus for the anthropology along with the notes, as well as I will also train you in terms of how to write the answer. So that's what we are starting on the 10th June, and this time we are launching the course in the online as well as in the offline mode. Our classes are going to be interesting, informative, and if you will be joining this course, at least for 2 to 3 years, you don't even need to update your notes, because I'm going to add upon the recent thing, the recent updates, as well as the case studies, examples, the new things, so that you will be feeling that something is being internalized in your mind, that you don't even need to stress upon remembering the things. Okay, so we are starting this course from 10th of June. You can join it, as well as you can get the basic details from the Google form that is available there. You can just fill it up, and the team is going to connect with you, as well as you can get in touch with the number that is available in the description box. Fine. So thank you so much for today. Now next class is going to be on the Friday. Thank you. Okay, examples in the aberration is going to be part for the next class.